Welcome to Dr. CM Edcast, your go-to resource for medical learning. Through discussions, case studies, tutorials, and debates, we help you learn, grow, serve.
You know, inside the nucleus of every single one of your cells, there's this constant, incredible process of information transfer going on. DNA gets transcribed into RNA, but that's not the end of the story. Not even close. That first RNA molecule is really just a rough draft, and it has to go on this amazing journey of processing before it's actually ready to deliver its message. So let's follow that journey together.
We're going to track a messenger RNA molecule from start to finish. We'll begin with its initial processing, then we'll see how it gets edited and refined. We'll also dive into how a single gene can create multiple messages, look at the parts that don't even get translated, see how these messages can be shut down, and finally, we'll touch on a really fascinating exception to the normal flow of genetic information.
All right, so first stop on our journey, post-transcriptional processing. This is basically where the cell takes that raw, freshly made RNA molecule and gets it ready for its super important job in making proteins.
Now, this first molecule is called a primary transcript. You might also hear it called heterogeneous nuclear RNA, or hnRNA. Just think of it as totally vulnerable and incomplete. The message is in there, sure, but it needs to be protected and cleaned up before it can even think about leaving the safety of the nucleus.
This whole makeover happens in three main stages. First, a special cap gets added to the front end, what we call the 5' end. Then, a really long tail is attached to the back, or the 3' end. And finally, the message itself gets a serious edit in a process we call splicing. Okay, this diagram here is perfect because it shows us the whole thing. You see the primary transcript peeling off the DNA. Then, boom, a 5' cap, which is a modified guanosine nucleotide, gets slapped on. This thing acts like a helmet, you know. It protects the RNA from breaking down and helps it hook up to the ribosome later. On the other end, you get this long string of adenine bases. That's the poly-A tail. This acts like a protective buffer, stopping enzymes from just chewing up the important message from the back. It's only after all this that the molecule is finally considered a mature mRNA ready to be shipped out. So with the protective gear on, the cell now turns its attention to the actual content of the message. This means it's time to cut out all the unnecessary bits to make sure the final instructions are crystal clear. So that primary transcript contains two different kinds of sequences. You've got exons and you've got introns. Exons are the expressed parts. They're the good stuff, the actual code that's going to become a protein. Introns, on the other hand, are intervening sequences. You can think of them as just genetic gibberish, basically, that's interrupting the important message. And they have to be removed, precisely. So how in the world does the cell pull off this kind of incredibly precise genetic surgery? Well, it uses a super sophisticated piece of molecular machinery called the spliceosome. It's this huge complex made of proteins and small nuclear RNAs, or snRNAs, and they all work together to find the introns, cut them out, and get rid of them. And this visual shows you exactly how it works. You have these little parts of the spliceosome called snRNPs, and their job is to recognize the boundaries between the exons and introns. They all gather on the intron, pull the two ends together to form a loop, and then the spliceosome just snips the intron out. It gets released in this unique lasso shape, which is called a lariat. Finally, it stitches the two exons together perfectly, creating one continuous, coherent message. It's pretty amazing. But the story of splicing gets even more interesting. Believe it or not, the cell doesn't always edit the message the same way. It can get creative, mixing and matching different exons to create totally different final messages, all from the same original gene. This leads to a pretty fundamental question, doesn't it? Can a single gene, just one stretch of DNA, actually be responsible for making more than one kind of protein? And the answer is a huge yes, all thanks to something called alternate splicing. By treating some exons as optional, sometimes keeping them, sometimes skipping them, the cell can create a whole family of related but distinct proteins from just one primary transcript. It's an incredibly powerful way to expand the functional diversity of our genome. And what's crucial here is just how efficient this whole system is. I mean, take the gene for a protein called alpha-tropomyosin. Through alternate splicing, a single transcript from this one gene can be processed to make 8 different mature mRNAs. Each one leads to a slightly different protein that's tailored for a specific type of cell. It's a fantastic example of genetic economy. Okay, but the story isn't over just because the splicing is done. Even in a mature mRNA molecule, there's more than just the protein coding sequence. At both ends, you've got these sections that don't get turned into protein, but trust me, they are far from useless. These are the 5' and 3' untranslated regions, or UTRs. They sit right before the start signal for making the protein and right after the stop signal. And these regions are packed with regulatory sequences that basically act like control dials. They can tweak how stable the mRNA is, how long it hangs around in the cell, and how efficiently it gets turned into protein. And just to show you how important UTRs are, abnormalities in these very regions have been linked to serious diseases, including breast cancer and Alzheimer's. So up to this point, we've been focused on creating and fine-tuning the message. But the cell also has some really powerful ways to just shut messages down. And one of the most elegant is a process called RNA interference, or RNAi. The RNA pathway is like a cellular security system. It gets triggered by double-stranded RNA, which is often a red flag for a viral infection. An enzyme called dicer finds this suspicious RNA and just chops it into little pieces called small interfering RNAs. These little pieces then get loaded into a protein complex called RISC. Now, armed with this guide, the RISC complex patrols the cell, hunting for any messenger RNA that matches that sequence. And when it finds a match, it destroys it. The gene is effectively silenced. This image here helps us zoom out and see the bigger picture of how an mRNA's fate is controlled. Once it's out in the cytoplasm, it's not on its own. It gets covered in all these different proteins and complexes that control where it goes, if it gets translated, and when it finally gets broken down. And that RISC complex we just talked about, it's a perfect example of this deep layer of control, where specialized machinery can target one specific message for destruction. All right, for our last topic, we're going to look at something that pretty much turns this whole process on its head. It's an enzyme that breaks one of the most fundamental rules of molecular biology. You know, the central dogma says that genetic information flows in one direction. From DNA, it's transcribed into RNA, and then RNA is translated into protein. But this enzyme, reverse transcriptase, does something wild. It takes an RNA template and uses it to build a new strand of DNA. It completely reverses the flow of information. This is the exact strategy used by retroviruses, and the most infamous example, of course, is HIV. As this diagram shows, the virus injects its RNA into a host cell. Then, using its own reverse transcriptase, it turns that viral RNA into DNA. This new viral DNA can then get shoved right into the host cell's own chromosomes, effectively hijacking the cell's machinery to pump out more viruses. It's a permanent infection, literally written into the host's genetic code. So, when you think about it, we've watched this RNA molecule go from a humble primary transcript to getting capped, tailed, and spliced. We've seen how it can be edited in different ways, how its non-coding parts control its destiny, and even how it can be targeted and silenced. It's so clear that RNA is way more than just a simple messenger. It's a dynamic editor, a precise regulator, an essential player in the whole intricate dance of life. Which really just leaves us with one final thought. What other incredible functions of this amazing molecule are we still yet to discover?